So hey everybody. So this far we've talked about basically how do we generate the raw sequence data. We talked about how do we go from a single sequence like a single sample sequence data to a genome, right? We talked about de novo genome assembly. And then more recently, we talked about in the midst of the pandemic, we can do align to reference and then pile up. So now let's put ourselves in the shoes of scientists who now have a bunch of COVID-19 genomes from all over the world. And now what do we actually do with that? So before this point, we've only been talking about, given one person's COVID-19 sample, how do I generate a genome sequence from it? Like, how do I actually analyze one sample? But now I'm asking, given every sample, so I think across the world, we have over like 1.2 million samples that have been sequenced so far. So given all of those sequences, what do I do with them? So yeah, today we're going to talk about aligning viral genome sequences. So here's a brief outline of what we're going to talk about today. I'm going to talk about different types of mutations. I'm going to talk about how we can think of mutations evolving down a phylogeny. And then I'm going to introduce this concept of sequence alignment, first with this pairwise sequence alignment, and then talk about multiple sequence alignment, which is really the focus of today's lecture. And then I'm going to end it with a more novel, like recent approach that people are using for multiple sequence alignment. So multiple sequence alignment is kind of the goal of today's lecture. Okay, so first let's talk about some types of mutations. So if you haven't taken a bio class, that's totally fine. Basically, the TLDR of everything you need to know about mutations is going to be covered on this slide. So basically, evolution is just mutations occurring. Like evolution and mutations are synonymous. So yeah, the most common type of mutation that we see is called a substitution mutation. Basically, given some DNA sequence or given some genomic sequence, one letter is replaced with a different letter. So here, T is substituted with G. Another type of mutation that occurs is an insertion, where one or more nucleotides are added. So here I had an original sequence ACT, and then over time, this new G was added into that sequence. So insertion, we're adding a new nucleotide. And then similarly, a deletion is when one or more nucleotides is deleted. So here the original sequence was ACGT, and then over time the G got deleted, and this was the result. So substitutions out of these three are by far the most common, but insertions and deletions do happen. So these mutations are heritable. So if some ancestral sequence encounters one of these mutations, any descendant of that sequence will inherit that mutation. So this is why, like with viruses, if a mutation happens in the virus of that's infecting a given person, and then that mutated virus then is transmitted to someone else, those mutations are inherited by whatever copies of that viral sequence are produced downstream. So yeah, so mutations are inherited down an evolutionary tree. Okay, so any questions about mutations? Hopefully you've heard of these before, or if you haven't, hopefully this makes sense. Any questions about these? Okay. So how do we think of mutations going down an evolutionary tree? Remember, an evolutionary tree is a tree structure where we have basically an ancestor, and then over time, forward in time is down, we have the leaves, which are present day sequences. So how can we imagine mutations occurring down a phylogeny? So here I have my ancestral sequence. And let's imagine that as time goes on, down this lineage of this evolutionary tree, maybe this G here mutated into a C. So this is a substitution mutation, right? So what this means is everything below this branch of the evolutionary tree is going to inherit this mutated C. So this is now the ancestral sequence of everything below this point. And likewise, maybe down this branch, this lineage, the G was deleted, right? So here we have a deletion of this G. So everything down this branch is going to inherit this deletion. So ACT will be the sequence down that branch. And I've drawn it as a gap just to kind of highlight that this is the deletion of this original G. But in practice, a deletion just means that we don't see it. So this sequence is actually just ACT. The G was just totally deleted. But yeah, I'm just putting a gap here, a dash, to help keep track of where it went. Okay, so now let's continue going forward in time. Let's say that from this point onward, this lineage didn't have any additional mutations. So the sequence that we would observe in present day from this person or this sample would be ACCT. It's the ancestral sequence, and then this one G turned into a C, and nothing else happened. Versus maybe down this lineage, maybe this A here mutated into a C. So this sequence inherited two mutations with respect to the common ancestor, right? It inherited this C and then this other C. And then maybe here, no mutations happened from this point onward down this lineage. So we just inherited ACT, right? We just inherited that deletion. And then maybe down here, maybe this C over here along this lineage transformed into a G. Okay, any questions about this hypothetical example? Kind of makes sense. Okay, so remember though, that in practice, we actually don't get to see the evolutionary tree or any of those mutated histories. All we can observe are the present day sequences. And actually, even this is misleading because remember, I put these gaps in here to help us keep track of the forward time evolution, but a deletion is just a letter got deleted, right? It disappears. So in practice, what we actually observe, if I were to sequence these 4 samples, are just these sequences here with no additional information, no context whatsoever. And what I want to do is try to basically see how are these sequences similar to each other, right? I don't have those gaps anymore. Oh, there's a question in the chat. So under the assumption that this continues on for a while, it's very possible to reach the original sequence. Yep, absolutely. So in general, it is theoretically possible for mutations to happen and then reverse. It's uncommon for the same position of a genome to mutate multiple times. So here, when I had this single position happen to mutate twice, this is actually a fairly uncommon thing to happen. And there's an assumption that we make usually called the infinite sites assumption that assumes that the genome is long enough that we have relatively mutations relatively happen at different sites. But with viruses, because viral genomes are so short, and because viruses mutate so quickly, positions that mutated once could mutate again. And yeah, as you mentioned, they could back mutate. They could revert to an older state. So yeah, absolutely, great insight. So yeah, it's possible, but not common. Any other questions about this? Cool. Okay. So that's basically how evolution happens down a tree, or how we at least model it happening down a tree. So now I want to introduce this concept of pairwise sequence alignment, and specifically, basically, so as you noticed, these sequences no longer line up. Like here I have ACCT and I have ACT. It's not obvious to me just looking at these two sequences that this T is the same T as this one. This C is the same C as this one, and this A is the same A as this one. It could be that this A maps to this A, this C maps to this C, and maybe this T maps to this C, but just a different mutation occurred down the line. So it's not clear to us what lines up with what, because I've deleted that information. So yeah, so pairwise sequence alignment is our approach of comparing two sequences to find what's known as homology, to figure out what letters of the sequence correspond to one another. So which of them was inherited from the same ancestral letter. So the computational problem here is your input are two sequences X and Y. So here I have ACT as X and I have ACGT as Y. And you'll see just by looking at this, they don't really line up. The A's and the C line up nicely, but this T doesn't line up with this G very nicely. And then there's an extra T on the end here. and there's an empty space here. Like there's nothing there at the end of the X. And then the output is a highest scoring alignment between X and Y. So here you'll notice if I just keep sequence Y the same, but I take sequence X and just plug in a little dash here. So I put a gap character. I just put a space between the C and the T. Now these two line up pretty nicely, right? 3 of their 4 positions are identical. Okay, so does anyone see any problems with how I formulated this? And if you haven't taken like a computer science class, maybe it's not super obvious, but I've basically introduced a term that I didn't define here. So what's the problem with this slide? Intuitively, the example makes sense, I think. The example, you go from, like you stick a gap here, and now they line up. Intuitively, looking at it, you can maybe solve this example, but I've introduced a term that I didn't define. Exactly, so as was mentioned in the chat, what makes it highest scoring? I've defined this highest scoring alignment, or I've mentioned it, but I didn't actually define what highest scoring means. So how do we actually score an alignment? Well, basically, we have to determine appropriate parameters for scoring a single column of an alignment. So a single column of a pairwise alignment is either going to be a match, right? So either we have 2 letters that are the exact same right next to each other, or it's a mismatch. So I have 1 letter and another letter, both in the same column, or it's a gap. I have 1 letter and that dash gap character. So in advance, I, as the biologist, have to think of what are appropriate parameters for a match, a mismatch, and a gap. I have to somehow pick these scores such that I want to give a good score to matches, and I want to give a bad score to mismatches or gaps. And I can kind of tweak these as I want. So maybe for whatever virus I'm looking at, maybe insertions and deletions are super, super, super unlikely. So maybe I want to make this gap penalty a really big negative number, versus maybe in other contexts, it's a part of the genome that inserts and deletes fairly easily. So maybe I want them closer to each other. So even defining this, even defining what is the appropriate value for a match, a mismatch, or a gap, this is a non-trivial thing that requires a decent amount of like biological insight. But for the sake of this argument, let's just pretend that someone who's an expert magically gave us the perfect parameters for each of these 3. So that tells me how to score a column of a pairwise alignment, just 1 position. And then the score of the overall pairwise alignment is the sum of the scores of the columns. So for example, let's imagine I had these 2 original sequences again, and let's say that this is one hypothetical alignment that I want to look at. So maybe take a second and think, what would be the score of this alignment? Any thoughts? Using this scoring convention that I've defined over here. What would be the score of this alignment in the middle? This one right here. Feel free to like type it in the chat or whatever you want to do. So I'm seeing a couple people are saying 0. Yeah, exactly. So let's take a look. So basically the sum, so the score of my overall alignment is the sum of the scores of the columns. So what's the score of this column? Well, this column I have A with A. That's a match. That's +1. Then I have a gap, right? I have dash with a C. So that's a gap. That's -1 according to my scoring scheme. Then I have a mismatch. I have C paired with G, which is also -1. And then I have T paired with T, a match, which is +1. So I have +1, -1, -1, +1. Overall, this is 0. Okay, so is this the highest scoring alignment between these 2 sequences, between X and Y, or can anyone think of something that's better? So here I have an alignment that has a score of 0. Yeah, so someone mentioned in the chat AC-T. So what if I put the dash after the C instead of before the C? Let's take a look. So the first column is a match, so that's +1. Second column is a match. That's also +1. Third column is a gap. Well, that's -1. And then last column is a match, so +1. So this one has +1, +1, -1, +1, or a score of 2. And it turns out that this is the best that we can do for these two sequences. Okay, does that kind of make sense? So now we've defined a scoring function for an alignment, and you can kind of now imagine that we do have a concrete computational problem here. Basically, out of all possible alignments that I could create, so out of all ways of putting gaps in either in either sequence, such that they end up being the same length, I want to pick the one that has the highest score, where this is the scoring convention that I'm using. Okay, any questions about that? Okay, so this computational problem, it turns out that there is a pretty efficient algorithm to solve it. I'm not going to go into the super extreme details of the algorithm. I'm just going to kind of give you a brief overview of how it works. And then if you're interested, feel free to reach out to me. I can send you some pointers. But basically, the algorithm that solves this pairwise alignment problem is called the Needleman-Wunsch algorithm. And the basic, so basically, this is an algorithm that's guaranteed to find the highest scoring pairwise alignment. So yeah, the details are pretty complex. If you're a CS major, or if you're thinking of doing a CS minor or anything like that, this algorithm is, I think, mentioned in CSE 101. So just kind of a call out if you're potentially pursuing that path. But basically, what happens is you first construct a matrix that has dimensions length of x plus 1, length of y plus 1. So here on the vertical axis, I have x, which is ACT, and then I have like this null character at the beginning. And on the horizontal axis, so the columns, I have y. So ACGT, but before it, I have this null character. So basically, you construct this matrix, and then you fill in this matrix using a recurrence relation that I'm not going to talk about in this class because it's super, super out of scope. But if you're interested, this technique is called dynamic programming. But again, this is covered in like an advanced computer science course. So I'm just going to kind of briefly skim over it. So using some recurrence relation that I'm not going to describe, you basically fill out this entire matrix slot by slot. And the details of how I'm coming up with these arrows and numbers, don't worry about it. I can send you some resources if you're curious about learning it. But somehow I have a relationship that lets me determine the value of each box based on the 3 boxes like above, diagonal to the left, and to the left of it. Okay, so I fill out this box. And then the last thing I do is I backtrack. So I follow arrows from the bottom right to the top left to reconstruct the alignment. How I do that from these arrows, again, don't worry about it, but basically I just follow arrows one by one and somehow cleverly reconstruct the sequence alignment. But the key takeaway of this, so I intentionally want to abstract away a lot of the details, but the key takeaway is that if we have some length k, so both of my sequences are roughly length k, this algorithm scales quadratically with the length of k. So you can imagine if I have k equals 100, I have to fill out a matrix that's roughly on the order of 100 squared. Versus if my sequences are length 1,000, my dimensions of my matrix are now 1,000 by 1,000. So I have to fill in a matrix that's roughly 1,000 squared. So the key takeaway is if I double the input size, so if I double the length of my sequences, I quadruple the amount of time it takes to fill out this matrix. Does that kind of make sense? So it scales quadratically with the length of my sequences. Does that kind of make sense? Any questions about that? Okay. And if you think of any questions, feel free to type them in the chat. Okay, so this helps me align 2 sequences, right? So if you gave me 2 COVID-19 genome sequences, I can now just plug them into this algorithm and out comes a pairwise alignment. But that wasn't my original question, right? My original question was I have 1.2 million COVID-19 sequences, and I want to align them all to each other. So this is a problem known as multiple sequence alignment. So remember, aligning two sequences where each sequence was length k was it was quadratic. It scaled quadratically with the length of k. So the number of operations I have to do, the time it takes, is some function of k squared, right? And for the case of COVID-19, the length of the genome is like 29,000. So we can think k is roughly 29,000. So it turns out that there's actually a very similar algorithm if I had three sequences. So instead of a pair of sequences, I had a triplet of sequences. I could do a similar thing that basically, instead of filling in a matrix, fills in a cube. And that would be, it would scale, cubically with respect to k. So if I have k equals 100, I would have to fill in this cube of 100 by 100 by 100. So this, if I double the number of sequences, I have to multiply the time it takes by 8, for example, like a cube, like it scales cubically. Any questions about what that means? Any concerns? Basically, each dimension that I add, it scales even worse by one more in that dimension. Kind of making sense? Okay, so basically, to align n sequences of length k, where again, k for COVID-19 is 29,000, n for COVID-19 is like over 1 billion. But yeah, the same approach would be big O of k to the n. So it would be scaling as some function, like the time it would take to run this algorithm would be some function of k raised to the power of n. So yeah, so plugging in COVID-19 numbers, k equals 29,000 roughly, n is around 1 million. Assuming that every single operation takes exactly 1 nanosecond, the number of nanoseconds needed to do 29,000 raised to the 1 millionth power, that's longer than the existence of the universe. So definitely that's not feasible, right? If we're trying to fight a pandemic, I can't write a program that takes longer than the entire universe has existed to finish running. Does that kind of make sense? Any questions about what that means? And this happens a lot with algorithms, that sometimes a naive or simple algorithm might be super, super inefficient and actually take like some unreasonably long theoretical amount of time to run. That would be like impossible to run within our lifetimes. So clearly we have to do something better. This is not fast enough to do any practical real-time public health intervention. So there's heuristics that exist that give approximate alignment. So the algorithm that I described, where for 2 sequences, I fill in a matrix, for 3 sequences, I fill in a cube, for n sequences, I fill in some n-dimensional, hypercube of some sort. That algorithm gives me an exact alignment. It's guaranteed to give me the optimal, absolute highest scoring alignment. So if I wanted to speed things up, I could use what's called a heuristic. So a heuristic is an algorithm that's not necessarily guaranteed to give me the absolute optimal answer, but it hopefully generally gives you a pretty good approximation. So it turns out that there are heuristics that scale on the order of quadratic with respect to the number of sequences times the length of the sequences, plus the length of the sequences squared times the number of sequences. So the number of operations performed is roughly n squared k plus k squared n. What that means, not too important, but the main takeaway is that it scales quadratically with the number of sequences rather than exponentially with the number of sequences. So much, much, much faster. And fortunately for us, we as the bioinformaticians, we as the public health people, don't have to worry about implementing this from scratch. Plenty of tools exist that do this. So these are 3 popular tools. MAFT is kind of the main one that people use nowadays. It was actually, out of these 3, it was the first one that was published. But since then, they've made a bunch of different iterations to it. So it's actually still generally considered the most accurate and the fastest. But yeah, there's multiple options. So basically what you would do is just take an existing tool that implements these heuristics for multiple sequence alignment. Okay, any questions? Kind of threw a lot of information at you. So if you have any questions, feel free. No, kind of making sense? Okay, so the last thing that I want to talk about, so prior to COVID-19, what I just described to you mapped these like multiple sequence alignment heuristic tools that scale quadratically with the number of sequences. Prior to COVID-19, those were the gold standard. That's what everybody would use to do HIV sequence analysis, Ebola sequence analysis, Zika virus, like whatever virus you're interested in, people would run a tool like MAFT to get a multiple sequence alignment in quadratic time. And the key difference, though, is that in those prior epidemics, we were dealing with like hundreds or maybe thousands of sequences. So scaling quadratically with respect to the number of sequences, that's totally fine. But it's unprecedented that in the COVID-19 pandemic, we actually, instead of being like on the order of thousands of sequences, we've now hit millions of sequences. So even those heuristics that used to be fast enough for these smaller epidemics, they no longer apply. And in general, in the future, as sequencing experiments get cheaper and cheaper, the hope is every single person that gets infected with some new virus, we should be able to sequence their viral genome. So in the future, this problem of scalability is just going to get worse. So we need something better than what we have with MAFT and like those other multiple sequence tools. So just like last lecture, where I mentioned that, hold up, we don't have to do this blindly. We have a high confidence reference genome that the other sequences, like every viral sample that I collect, it's going to be different from the reference, but it's going to be extremely, extremely similar. Like any differences are going to be fairly minor. So the same logic as last time holds here. Is there any way that I can leverage this reference genome to somehow take advantage of these similarities that all of my other sequences have with respect to it to speed things up? So on this slide, let's imagine that this green thicker line is my reference genome for COVID-19. And then each of these other colored lines, the red, orange, purple, and blue lines, these are all consensus sequences that I used last week's lecture to build that I now want to do multiple sequence alignment. So I want to align, let me draw on my screen. I want to align these sequences with each other in a multiple sequence alignment, but I have access to a reference genome that I know in advance all of these sequences are super similar to. So remember that pairwise alignment, it scales quadratically with respect to the length of the sequences, but here I'm fixing the length. The length is 29,000 for all of these cases. But what I want to avoid is scaling exponentially with respect to the number of sequences, right? This is n, this is the number of sequences, and I want to avoid scaling exponentially with respect to that. So what if instead of aligning my sequences with each other, because I have this high confidence reference genome, what if I align each of my consensus sequences pairwise with the reference genome? So I do the red sequence pairwise aligned with the reference genome. And independently, what if I do the orange sequence pairwise aligned with the green genome? And also independently, the purple genome pairwise aligned to the reference genome, so on and so forth. Okay, does that starting point kind of make sense? So basically, let me do a pairwise alignment of every single one of my consensus sequences against the reference genome. Okay, and then what I can do is let me use the positions of my reference genome. So here, every one of these little squares is one of the letters of my reference genome. It's either an A, C, G, or T. But remember that in those pairwise alignments that I did, every letter in my consensus sequences aligned with one of the letters of my reference genome. So what if I use my reference genome's letters as anchors to combine all of the consensus sequence letters that align to that letter? So let's look at the first letter first. So this is the first letter of my reference genome. What if I take the first letter or whatever letter of my red genome that happened to align to the first letter of my reference, and also take whatever letter of my second genome that happened to align to the first letter of my reference, versus let me take whatever letter in my purple genome aligned to the first letter of my reference, etc. So given the first letter of my reference genome, let me extract whichever letter from each of my consensus sequences happened to align with that letter. So after the alignment, happened to be in the same column as that. And then I can do the same thing for the second position, the same thing for the third position, same thing for the fourth position, so on. And basically, I can create a multiple sequence alignment where the columns of the multiple sequence alignment are simply the columns of my reference genome. And each value in any given column of the multiple sequence alignment is just whichever letter of each sequence happened to have a pairwise alignment to that letter of the reference genome. Does the approach kind of make sense? Any questions about that? Throwing a lot at you, so feel free to ask questions if you have any. Oh, so there's a question in the chat. So this would take n times k squared time. Exactly. So remember the other heuristics were n squared k plus k squared n. But this would take just k squared n. So just k squared to do any of these pairwise alignments. And then I would do it n times. So it scales linearly with the number of sequences I have. So there was a question in the chat, would you use different reference genomes for different strains? So I don't know if you're talking about COVID-19 specifically, but if you are talking about COVID-19, oh yeah. So for COVID-19, actually, I don't like that in the news world, they're using the term strain for this, because they're different lineages of COVID-19, but they're all still almost identical to the reference genome. So actually that single reference genome that I showed you last time, you would use it for every single COVID sequence that you get, because they're all still 99.9% identical to the reference. The whole notion of strain, I think I don't know what the official definition of a strain is, but yeah, in COVID-19, it's kind of a misused term. But yeah, they're all still pretty much identical to the reference. Yeah, so the news, so in general, I've had a lot of problems with the reporting of the news during COVID-19 pandemic, because things that are not super surprising or not super crazy, the news then makes an article that says oh, the new UK variant has now spread through all of the US. I don't know. It yeah, yeah. I have feelings about that. But yeah, so they're actually all very, very, very similar looking sequences. It's just that they happen to have key mutations in specific parts that are potentially problematic. So for example, we mentioned a few times ago that there's that spike protein. That's how, if I have a cell and I have a little coronavirus that has all these little proteins on it, these spike proteins are kind of the key by which the coronavirus invades the cell. And the antibodies that we produce as I don't know if anyone here has gotten the vaccine yet, but basically the vaccine, one of the parts of the virus that it's targeting is the spike protein. So you develop antibodies to the specific version of the spike protein that they tried to design the vaccine to look like, which means that if the spike protein happens to be maybe this window of the reference genome, and then all of a sudden new mutations happen in some new lineage of COVID-19 somewhere in the world that significantly enough changes the shape of the spike protein to no longer look like what the antibodies are expecting, that would be very bad. But it would still, as a global just sequence comparison standpoint, the sequence as a whole still looks almost identical to the reference genome. It's just that these differences happen to be very biologically meaningful. Does that kind of make sense? That's a great question, by the way. Yeah, and feel free to interject with any more questions. That was great. Yeah, no problem. Cool. Okay, so we've outlined a general approach, but how do we actually implement it? And this was actually, if we rewind roughly 1 year ago, this was an idea that I had to try to speed things up. So so first let's talk about some of the benefits of this approach. So first of all, these pairwise alignments against the reference are done completely independently. So what's nice is that there's a ton of room for parallelization. So this is something that if you end up taking any computer science classes later on, the notion of how do we parallelize something is an important computational question. And if you think about it, and the analogy that I like to use is for cooking. Like imagine that you're trying to cook something. If you have two people that are cooking together, there might be some tasks that you can distribute to different people. So maybe like chopping the onion and boiling the water, two different people can be doing that at the same time. But like chopping the onion and then chopping the carrots, if you only have like one knife and cutting board, it doesn't matter if you have 5 different people. Only one person can do that, like those tasks at any given time. So kind of the tie-in to algorithms in computer science is that some computational problems can be broken down into independent subproblems that can be worked on simultaneously, just like some cooking recipes can be broken down into steps that different people can work on simultaneously, but other ones cannot. Sometimes there's dependencies that you cannot do them independently from one another. But for this, beautifully, each of those pairwise alignments is completely unrelated to the other ones. So if I have a million sequences, suppose like theoretically, I could have a million of these pairwise alignments going at the same time if I had some computer hardware that could handle that. Would this be asynchronous versus asynchronized versus synchronized? So in this case, the pairwise alignments are completely independent. So they could be done at the same time, or they could be done separately asynchronously. I don't know if that answers the question, but in this case, you would probably want them to be done at the same exact time, just to kind of lessen your runtime overall, if that kind of makes sense. Cool, nice. So that's very nice, right? So I can parallelize massively. So if I get a computer that has like 128 cores in it, I can throw all 128 cores at this problem at the same time. And the second benefit is that it scales linearly as a function of the number of sequences. So remember, it scales quadratically as a function of the length of the sequence, but the length is fixed. The COVID-19 genome is 29,000. That's a constant, right? Maybe it's kind of big, but it's still fixed. Versus the thing that changes as the pandemic goes on is the number of sequences. And this scales linearly. If you double the number of sequences, you double the runtime, which is very nice. You generally can't get better than linear for this type of like data analysis type problem. So yeah, so this is much more feasible as the data set size grows. Okay, so one of the limitations of this is that it's difficult to handle insertions with respect to the reference genome. So I showed you that basically to take all of our pairwise alignments and concatenate them into a single multiple sequence alignment, I used each position of the reference genome as an anchor. But the problem with that is, what if I had an insertion with respect to the reference genome? So what if there was, in one of my alignments, a gap in the reference genome? Unfortunately, I have no way of consolidating insertions with respect to the reference genome. So for the purposes, so for the purposes of this, in general, you just kind of throw out those insertions. So you can keep matches, mismatches, and deletions with respect to the reference genome, but not insertions with respect to the reference genome. So, and again, an insertion with respect to the reference genome is a letter that shows up in the, like one of your collected samples, an insertion that doesn't show up. So like ACT in your collected sample versus AT in your reference genome, that we cannot handle. But the other way around, so ACT in your reference genome versus AT in your sample, that's a deletion with respect to the reference genome. That one we can handle fine. So yeah, so generally these types of aligned to reference approaches, you just throw away insertions with respect to the reference. But fortunately, for any type of downstream analysis that we want to do, so I'm going to talk about molecular epidemiology in the next few weeks and like phylogenetic inference. Fortunately for us, these insertions with respect to the reference genome are fairly unique, and as a result, don't tell us very much relationship information between sequences, because usually only one sequence or a handful of sequences will have this insertion. So throwing it out doesn't throw out that much information that we can use. And also, if we wanted to, we could theoretically restructure our reference genome to be representative of any common insertions that we expect to see. Right? So if we could just add more stuff to the reference genome, more of our sequences are going to have deletions with respect to the reference, but that's fine. So this is actually a fairly simple issue to circumvent. Okay, so now this is kind of tying back to something that I worked on last summer, right when the COVID-19 pandemic was first starting. But basically, remember a couple weeks ago, I taught you about this read mapping problem, where I have a bunch of short sequences that I'm mapping to a single reference genome, right? So my input was a reference genome. And in this case, let's pretend I have what are called long reads. So instead of length 100, let's imagine my reads are pretty long. And then my output is an alignment of each sequence, each of my reads against the reference genome. So exactly the read mapping problem that we talked about a few weeks ago, this is basically what the read mapping problem was. Does that kind of make sense? So basically, this goal of taking every one of my query sequences, each of my consensus sequences, and pairwise aligning them against the reference genome, that is essentially exactly the read mapping problem. Just our reads are super long. Does that connection make sense? Any questions about this? No. So does anyone remember what tool I mentioned is kind of like the fanciest tool for doing this right now, like the read mapping? It's the fastest tool. It's like really nicely coded. I don't know if anyone remembers. No, that's okay. So basically, my thought process was I don't want to do any more work than I have to. So let me come up with a tool that can leverage existing tools that do read mapping super well to hopefully try to speed up this problem. So I came up with a tool called Viral MSA that just wraps around existing read mappers to perform this exact reference guided multiple sequence alignment technique. So I wrap around a few different ones that we talked about a couple weeks ago, and so this was the answer to my question. So I tested it with a bunch of different ones, and Minimap2, which is like this amazingly written tool, consistently, it gave me the best speed and the best accuracy. So basically, I implement a bunch of different tools as desired, just to show that it's flexible, so that like in the future, if Minimap2 is no longer the hottest tool, if something better comes out in the future, I can just wrap around that instead. But for today's goals, just using Minimap2 is the best. So you can find it on GitHub right here. I can pull up the GitHub repo later if you want. And I published it, last summer. So basically, one of the key benefits of this is that, so on the COVID-19 data set of roughly 100,000 sequences, this was back a year ago, so we were only at 100,000 back then. MAFT would take like 3 to 4 hours to run, at least. Like it would be on the order of hours for sure, maybe days. Versus this approach of basically using Minimap2 to align each of the consensus sequences against the reference and then concatenating them together on 100,000 sequences took like less than a minute on my laptop, which is awesome. Because now problem solved, right? If we have 1 million sequences, it'll take me maybe 10 minutes on my laptop. But the beauty is I can parallelize Minimap2 as much as I want. So if it ends up being that my 8-core laptop ends up being too weak, I can always throw like some 128-core machine at it and just like throw all the compute resources I want. So problem solved. I'll pull up the Viral MSA repo and I'll post it in the chat. So feel free to check it out. It's just a single script. It's a little long, but like not too bad considering. But basically what it does is I've predefined some reference genomes that I think are kind of the good ones to use. And then basically, where am I? So you basically parse the user arguments, and then you run, oh yeah, you download the reference genome and like set it up. You run the aligner, so in this case, minimap2, and then you parse its output and just concatenate it into a multiple sequence alignment. And then the concatenation step happens, where was it? Here. This is where I like convert from the read mapping to the multiple sequence alignment. So yeah, if you're interested, feel free to take a look at this. And yeah. Cool. So any questions? That's all I had to discuss. But any questions from anyone? Any concerns? Anything like that? Any questions? No? Cool, okay. Well, yeah, we can call it there then. If you have any questions about this stuff, so this is a problem that I'm like pretty interested in. So as you're like reading up on stuff, if you find it interesting, feel free to reach out to me. I'm happy to answer any questions. But cool. Yeah, okay, well, I'll call it there then. Thanks everyone for coming, and I will see you all next week. Bye, everybody.